A researcher in a sealed lab in Uppsala, Sweden, leans over a fragment of 2,000-year-old parchment, and instead of reading a single word of the ancient Hebrew written across its surface, she scrapes a few invisible grains of dust off the blank, unwritten back. That dust is not old ink. It is not stone. It is skin, animal skin, and buried inside it, if anything survived 2,000 years in a cave, is DNA. 7 years later, that dust would help dismantle an assumption scholars had built an entire chapter of biblical history around. Somewhere in that same 7-year project, a team would end up testing not just scrolls, but sandals and water skins, and scraps nobody had ever thought worth analyzing before, treating every piece of leather recovered from those caves as a potential witness that had simply never been asked the right question. And one of the strangest results wouldn't come from the sacred text at all. It would come from a pair of ordinary drinking pouches and from a single word of scripture that turned out to be sitting on the wrong animal entirely. This is the story of what happened when a molecular biologist who studies microscopic worms sat down next to a scholar of ancient Hebrew literature on a bus during a new faculty orientation at Tel Aviv University, and decided, almost on a whim, to try something nobody had fully attempted before: sequencing the genetic code hidden inside the most famous religious manuscripts on Earth. The team behind this was not improvising. It was led by Professor Oded Richavi and Professor Noam Mizrahi at Tel Aviv University, working alongside Professor Matthias Jakobsen at Uppsala University in Sweden, Professor Christopher Mason at Weill Cornell Medicine in New York, and the Israel Famous St. Wyatt Antiquities Authority, which holds custody of the scrolls themselves. Their findings were published in June 2020 in the journal Cell, one of the most demanding peer-reviewed venues in all of biology. After 7 years of work extracting, sequencing, and cross-checking genetic material almost too degraded to read. This wasn't the first attempt to pull biological information out of the scrolls. Scientists had shown, as far back as the year 2000, that it was technically possible to amplify small traces of animal DNA from Dead Sea Scroll fragments using PCR, the same basic amplification technique used in everyday genetic testing. But PCR-based methods could only answer very narrow questions, essentially confirming a single species at a time from a single tiny sample. What changed by the time this team began its work was the rise of deep sequencing, a far more powerful approach capable of reconstructing much larger stretches of genetic material, even from samples this degraded, building on techniques recent studies had begun applying to historical parchment and vellum elsewhere in Europe. That technological gap, the difference between merely detecting DNA and actually being able to compare and relate it across dozens of specimens, is what made this project possible in a way it simply wasn't 20 years earlier. Here's a way to picture what they were actually doing, because sequencing ancient parchment sounds abstract until you see the logic underneath it. Every piece of parchment used to write the Dead Sea Scrolls started life as an animal hide, a sheep, a cow, sometimes a goat. Think of each hide the way you'd think of a shipping label on a package today, a barcode stitched invisibly into the material itself, one that can tell you not just what animal it came from, but which other pieces of parchment came from a related animal, the same flock, potentially the same herd, even the same individual. Ink can be forged. Handwriting can be imitated. A genetic signature embedded in the skin itself cannot. And unlike the text on the front of a scroll, which every scribe, every scholar, and every forger across 2,000 years has had the chance to study, copy, and argue about, the biological identity of the material underneath was invisible to every single one of them. Nobody in antiquity was choosing parchment based on what its DNA would someday reveal. That is precisely what makes it such an unusually honest witness. The first job was simply figuring out what animals the scrolls were even made from, something nobody had ever confirmed with certainty at this scale. Working from 26 scroll fragments plus related leather objects like sandals and ancient waterskins, the team found that the overwhelming majority, 24 of 26 tested fragments, were written on sheepskin. Only 2 came from cattle. That imbalance itself tells you something practical about how these communities operated. Sheep and goats were the animals actually suited to the desert economy around Qumran, easy to graze on sparse vegetation, requiring far less water than cattle to survive. If the scrolls had been produced entirely locally, you'd expect the parchment to reflect that reality almost universally. Mostly, it did. It's the exceptions that turned out to matter. Quickly, before going further, here's what that simple species count actually sets up. 2 out of 26 doesn't sound significant on its own. It's a small number. But once you understand where these scrolls were physically written, that small number turns into one of the most important clues in the entire study. Qumran, the site near the Dead Sea where most of the scrolls were discovered, sits in one of the driest, harshest environments in the entire Judean desert. Cattle need something sheep and goats mostly don't: large amounts of grass and standing water, resources that simply do not exist in that landscape in any reliable quantity. So when the DNA came back cow instead of sheep, it wasn't just a footnote about the animal on the label. It was a geographic fingerprint. A cowhide fragment sitting in a Qumran cave almost certainly did not start its life anywhere near Qumran. Someone brought it there from somewhere else, somewhere wetter, somewhere greener. That single insight is what turned a straightforward species identification project into something far stranger. And this is where the results stop being a footnote and start being the headline. Scholars had long debated whether 3 separate fragments of the Book of Jeremiah, found among the scrolls, actually belong to the same original manuscript. It's an easy assumption to make. They're the same book, found in roughly the same place, written in a similar hand. Historians have been treating them as pieces of one shattered document for decades. Then the DNA came back. One of the 3 Jeremiah fragments was made from cow skin. The other 2 were sheep, and that difference wasn't cosmetic. According to Professor Mizrahi, fragments made from genetically unrelated animals, especially animals from entirely different species, are extremely unlikely to have come from the same original scroll. A scribe assembling a single manuscript used consistent material, typically hides from animals raised together, processed together, prepared together. A cowhide fragment and 2 sheepskin fragments almost certainly started as 3 separate pieces of parchment, likely made in different places, possibly written by different scribes, and only later grouped together by scholars who had no way of knowing the material itself told a different story. Sit with that for a second, because it means one of the foundational assumptions built into how the Dead Sea Scrolls have been studied for 70 years, that certain fragments obviously belong together simply because they contain the same biblical text, doesn't hold up once you can actually test it. Generations of scholars had made a perfectly reasonable inference from the evidence available to them at the time. It just turned out there was another kind of evidence sitting invisibly in the material the whole time that nobody had a way to read until now. In the next part of the study, the team pushed even further than species identification. Using both mitochondrial DNA, inherited only through the mother's line, and nuclear DNA, inherited from both parents, they compared individual sheep genomes against each other, looking for close relationships between the specific animals whose skins ended up as scroll material. Fragments made from closely related sheep were treated as more likely to share a common origin, potentially the same production batch, the same flock, the same moment in time. Fragments from distantly related sheep, or from an entirely different species altogether, were treated as more likely to represent separate origins, separate sources, separate histories. That method also let the team solve smaller physical puzzles that had nothing to do with scrolls at all. Among the leather objects tested were 2 ancient water skins recovered from the Qumran caves, cataloged only by inventory numbers, their relationship to each other unknown. Genetic testing matched one specimen directly to the other, confirming they came from related material, quietly closing a much smaller mystery sitting right alongside the much bigger one. The team also tested ASE of ancient sandals recovered from the same desert sites, one likely made of goat skin, the other from an animal source. The DNA couldn't confidently pin down at all. A reminder that even with modern sequencing technology, some of what the desert took 2,000 years ago simply isn't coming back. While the Tel Aviv and Uppsala labs were focused on the animal DNA itself, Christopher Mason's lab in New York was running an entirely separate analysis on the same specimens, looking not at sheep or cow genomes, but at the scroll's microbial contaminants, the bacteria, fungi, and other microscopic hitchhikers that had colonized the parchment over 2,000 years of storage and handling. That metagenomic side of the project served 2 purposes at once. It helped the team distinguish genuine ancient signal from modern biological noise, and it opened a separate line of inquiry into the scroll's conservation, since knowing exactly what kind of microbial life is currently living on a 2,000-year-old document matters enormously for anyone trying to keep it intact for the next 2,000 years. Here's the part that made this project harder than almost any other ancient DNA study attempted up to that point. 2,000 years is an enormous amount of time for genetic material to survive, and the Judean desert, hot and only occasionally protected by cave conditions, is not a gentle environment for it. The team recovered DNA at extraordinarily low coverage, in some specimens averaging roughly 1/100th of a full genome's worth of readable material, pieced together from 286 separate sequencing libraries. On top of that, every scroll had been handled by archaeologists, curators, and researchers for decades after its rediscovery, layering modern human DNA directly on top of whatever ancient animal DNA remained. Before the team could analyze anything about the sheep or the cows, they first had to computationally strip away generations of accidental human contamination, fingerprint by fingerprint, just to get back down to the signal they were actually looking for. There's a particular technical trick worth understanding here, because it's what let the researchers trust their results at all, despite how little genetic material survived. Ancient DNA degrades in a very specific, predictable pattern over time, a slow chemical process where certain bases at the ends of DNA fragments convert into other bases at a measurable characteristic rate. Modern contaminating DNA, by contrast, doesn't show that pattern because it hasn't had 2,000 years to degrade. By checking every sample for that telltale signature of age-related decay, the team could tell the difference between DNA that had genuinely been sitting in that parchment since antiquity and DNA that had been deposited far more recently by a curator fingertip. It's the genetic equivalent of checking whether a photograph has yellowed with age before trusting its timestamp. Several of their samples showed exactly the decay pattern old DNA should show. A few others, revealingly, showed a pattern more consistent with modern contamination or recent handling damage, and the team flagged those honestly rather than treating every result as equally reliable. Let's quickly restack everything before the last piece lands, because it changes what all of this actually means. 24 sheep, 2 cows out of 26 tested. Fragments. Cattle that almost certainly couldn't have been raised locally at Qumran. Jeremiah fragments long assumed to be one shattered scroll, revealed by species alone to likely be 3 separate ones. And underneath all of it, a technique sensitive enough to match 2 unlabeled water skins to each other purely from degraded genetic residue, and precise enough to separate genuine 2,000-year-old signal from a curator's fingerprint left decades ago. Put together, those results point towards something bigger than any single fragment. The Dead Sea Scrolls have often been described in popular retellings as the private library of one isolated religious community, the Essenes, writing and copying texts in relative seclusion at Qumran and nowhere else. That picture comes largely from ancient writers like Josephus and Pliny the Elder, who described an ascetic Jewish sect living apart from mainstream society near the Dead Sea, a description scholars connected to the Qumran ruins once the scrolls were discovered nearby between 1947 and 1956. It's a compelling, tidy narrative. One community, one library, one worldview, preserved in amber for 2 millennia. The genetic evidence complicates that picture considerably. If some of the parchment used cattle that could only have been raised somewhere with real grass and real water, somewhere well outside the desert, then at least part of what ended up in those caves wasn't produced on site. It was brought in from other Jewish communities, possibly from significant distances away, and folded into the collection alongside material that genuinely was local. That reframes the scrolls themselves subtly but significantly. Instead of one sealed homogeneous archive written entirely by one group in one place, the DNA suggests something closer to a gathered collection. Texts pulled together from multiple sources, multiple communities, multiple points of origin, all eventually stored in the same set of caves. Some scholars have raised versions of this idea before on purely textual grounds, arguing the diversity of viewpoints across the scrolls looks less like the output of a single insular sect and more like a broader cross-section of Second Temple era Judaism. Genetic evidence doesn't prove that broader theory outright, but it does provide a completely independent line of physical evidence pointing in the same direction, which is exactly the kind of convergence that makes a historical argument harder to dismiss. It doesn't erase the Essene connection to Qumran, which rests on decades of separate archaeological and textual evidence. It adds a layer underneath it that no amount of paleography or ink analysis could have revealed on its own. So here is the honest, careful version of where this actually leaves things, because a finding this significant deserves the caveats as much as it deserves the headline. The DNA coverage recovered from most specimens was extremely thin, averaging closer to 1% of a full genome in many samples, which means that genetic relationships the team identified are probabilistic, not absolute certainties. Saying 2 fragments likely came from related animals is a strong statistical inference built from careful modeling. It is not the same as a courtroom-grade genetic match. The conclusion that cowhide fragments originated outside Qumran is also, ultimately, an ecological argument rather than a direct geographic one. The DNA identifies the species, and researchers infer the likely origin from what is known about where cattle could plausibly have been raised in that period, not from a genetic marker that says made in Galilee outright. Someone could, in principle, have transported feed and water to raise cattle near Qumran at real cost and effort, even if it was unusual and archaeologically unattested. The inference is a strong probability built on regional ecology, not a certainty ruled out by physics. It's also worth being precise about scale. This study examined only 26 scrolls, fragments plus a handful of related leather objects, out of a collection that spans roughly 900 manuscripts recovered from 11 different caves near Qumran, alongside related material from other Judean desert sites like Masada. What applies to the specific fragments tested here shouldn't be assumed to apply uniformly to every scroll in every cave. The researchers themselves were explicit that this was as much a proof of concept for the method itself as it was a final answer about the whole collection's origins. Future sampling, larger sample sizes, and future improvements in sequencing sensitivity could sharpen this picture considerably, confirm patterns seen here across a much larger sample, or complicate them further in ways nobody can predict yet. And it's worth remembering that a shared species or a close genetic relationship between two fragments is evidence of common origin, not proof. It raises or lowers probability rather than settling a question outright, the same way a fingerprint match strengthens a case without being the only piece of evidence a historian would want before rewriting a chapter of scroll scholarship built up over 70 years of careful, painstaking work. If the idea of reading history off degraded animal skin instead of the words written on it is the kind of detective work that hooks you, this channel has more of exactly this waiting, and subscribing means you'll catch the next one. What the scroll dust in that Uppsala lab ultimately gave back after 7 years of work wasn't a single dramatic discovery so much as a quiet correction to how confidently anyone should claim to know where an ancient document actually came from, just by looking at the words on its face. 2 fragments of Jeremiah, written in careful, deliberate script sitting for 2,000 years under the assumption that they were always one document, torn apart not by an argument or a new translation, but by a few invisible flecks of skin cells scraped from the blank side nobody had ever thought to test. It's worth sitting for a moment with how strange that reversal actually is. For most of the Dead Sea Scrolls' modern history, the tools available to scholars were paleography, comparing letter forms and handwriting styles, and textual criticism, comparing the actual wording against other known versions of a book. Both are careful, rigorous disciplines, built up over generations of expertise, and both have their own long track record of resolving genuine historical disputes on their own terms. But both, in the end, only ever look at what a scribe deliberately wrote. Neither could ever have detected which animal a piece of parchment came from, because that information was never meant to be read by anyone. It was simply there, embedded in the material by biology rather than by intention, waiting 2,000 years for a technology that didn't exist yet. That is the real shift this study represents, not just a new answer to an old question about Jeremiah, but an entirely new category of evidence sitting quietly underneath every other ancient manuscript ever recovered, ready to be asked questions nobody thought to ask before. Tell me in the comments which detail struck you more, that the Jeremiah fragments weren't actually the same scroll after all, or that a 2,000-year-old waterskin could still be genetically matched to its twin, decades after anyone last thought to ask.